welcome to another episode of Next Gen Scientist. I am your host, Ana Rita Cavaco, and as always, I'm here to bring the best of the science done here at ISTA and the stories of the people who do it. And today, it's my great pleasure to have on the purple couch Marina Osorio, a plant biologist who is a postdoctoral researcher at the group led by Professor Eva Benkova. Hi Marina,
Hello.
Thank you so much for joining us today and for
welcome.
sharing with us your work towards becoming a plant biologist.
Yes, no worries.
Now, before you tell us everything about your work as a plant biologist and how you became one, let's start with our round of 5 quick questions.
Okay,
So, ready?
I hope so.
One word that you use to describe your research.
It's challenging.
What is your brain fuel? Caffeine, snacks, memes, or panic?
Early in the morning, caffeine, and then during the day, snacks, sugar. Yeah, for sure.
Very well. What's the most unexpected place your research has taken you?
Unexpected. I'm not sure about unexpected, but through my research, I was able to already travel through most continents in the world, and most importantly, connect with different people from all over. So I think that's the most interesting part as well.
That's really cool.
Yeah.
So because of science, you've been all over.
Yeah. Conferences, internships, or different jobs, really.
That's really cool.
Yeah.
And are you team night owl or early bird?
I think I'm neither. I would much prefer to work like consistently through the day and to try to keep my endurance levels, because I feel like sometimes science can be like a marathon, like a very long one.
That's very true. There are periods.
Yeah.
And complete the sentence. I never thought being a scientist would mean...
To be a professional project manager. I think, as you hear later, I really felt like I have this scientist feels in me since I'm very young. And when you're young, you think a scientist is that image of like the naturalist in the 1800s or whatever, and it's like you're just gonna wander around and you're gonna observe nature, you're gonna do whatever you want and like take a long time to do your project, but you don't really care about managing everything. But actually the reality is that we have to manage. We have to manage a lot of things, different things happening at the same time, and we have to make sure that we deliver, right? So that's
Exactly.
Yeah.
Very well.
Yeah.
Now, a few weeks ago, we met in your lab, and I had the opportunity to follow an experiment that you did for the course of 3 weeks.
Yeah.
So you showed me how different soil conditions affect the growth of plants and their nutrition. Would you like to build up on this before we show the experiment to our viewers?
Yeah, that's fine. So usually I suppose you imagine that just like us, plants, they need nutrients to grow and to function. And these nutrients are most often present in soil. There are 3 different, the main nutrients are nitrogen, phosphorus, and potassium, and they are not very well spread or distributed in the soil. There will be different patches which will have more of something. And also with nitrogen, there are different forms, different chemical forms that plants can take, or some plants prefer one form, some plants prefer another. So for example, that's the case of nitrate and ammonium. And I'm working with Arabidopsis thaliana. Arabidopsis thaliana, or Theocris, is this model plant that we use in the lab all the time. It's very boring. It's not as interesting as some trees that you see outside or some crops, but it's very easy to work with, and we use this as the basis for like all plant projects. And then, of course, you can build up and do some more interesting plants. But Arabidopsis, it really doesn't like ammonium so much as a nitrogen source. It likes more nitrate. And the nitrate in soil is more distributed in the lower layers, like around the bottom of the soil. Whereas the ammonium is usually fixed a bit more in the topsoil. And so I am looking in my experiment how different genotypes of plants, so plants with different genes where like one gene maybe is functioning 100% or maybe the other plant doesn't have this gene functioning. And to see if this gene is involved in how plants take nitrogen, then I would see how does it behave in this type of soil that we have. So
Very
that's
well.
more or less.
Very well. So when you say nitrate and ammonia,
Yeah.
we are talking about different molecules that have nitrogen on them,
Yeah.
which is important for plant nutrition, right?
Mm-hmm. Yeah, exactly. And just one thing that I think it's very important to say, because how, like, if you think of plants, they cannot run away, of course, right? They're like fixed to soil. They don't have legs. And so they have to be very quick to respond to their environment. And that's the same with the below ground, not only with the above ground, but below ground, they have to be very quick in sensing what's happening and in communicating this signal through the plant, right? So some molecules which are very important in helping this sensing and transmitting the signal, this communication network, are hormones. So plant hormones, for example, we have auxin, cytokinin, salicylic acid, ABA, we have different types of hormones in plants, and they are the messenger molecules that will help the plant understanding what's happening, also below the ground.
Very cool. So
Yeah.
let's take a look into this experiment that Marina showed me, and we'll be right back.
Enjoy.
So today what I'm doing is preparing the experiment. So now my plants are ready to go into this incubator. So I've set the temperature to around 21 degrees, which is what Arabidopsis plants are usually happy with. And we have a long day, like mimicking summer days. And so that will make them grow quite nice. And what I have to do here, you see I have my mutant genotype and I have my wild type. And so we're going to have to wait a few weeks to see how the experiment progresses, how the roots progress.
So what we have here is a wild-type plant where all the plant genes are functional, including the ones which allow the plant to scavenge, to sense and scavenge for nitrogen. As you can see, the root is growing quite well to the bottom of the plate. It's also growing a bit sideways. And we can compare this plant to a mutant plant, which I will show you here. So this mutant doesn't have one gene functioning, and that means the root system's architecture has changed completely. So this plant is not so efficient in taking nitrogen, in sensing nitrogen and taking nitrogen, and that means also that the shoot will grow smaller and it will produce less fruits, less seeds. Something that is very important to plants is what's below the ground, and that's the roots. So if you look here in our experiment, what we can see, the shoot, the leaves, they have some area, and they are the part responsible for photosynthesis, right, in the plant. But the root often can have a much bigger area, and it can cover much more ground than the shoot. And so why does the root need to have such big area under the soil, what we don't see? Is because plants, they, even though they do photosynthesis with their leaves, with their shoots, the green part, they also need nutrients, just like we do. Because taking nutrients from soil is energy expensive for plants, they will only take as much as what they need in order to grow strong. And so what happens in agriculture often is that the fertilizers which are added to the farms are added in excess. They are added too much. And then because the plant doesn't take it all, the remaining fertilizer ends up polluting the environment. And that's the reason why what I'm trying to understand is how plants sense the nitrogen availability in soil. And so plants which are better able to sense the nitrogen source available in the soil, they will, the roots will grow more towards this source, and they will be able to take up more, metabolize this nitrogen better, And that means that in their leaves, they will grow more. And that means also better photosynthesis. That means the plant will be more productive. When it comes to producing fruits or seeds, it will usually produce more. So usually there will be different genes, right, that control different traits of the plant. And this nitrogen sensing and nitrogen use efficiency is also something that is controlled by different genes. So now it's been 21 days or 3 weeks since I started the experiment in soil, if you remember, after that transfer. And we have here the different genotypes, right? We have the wild type plant. The wild type means a plant where the gene is functional. And we have the mutant plant. So the mutant plant is a plant where the gene is not working anymore. And what we see in this case is that the wild type plant is able to sense the nutrient availability in the soil. And in this case, as I told you before, with nitrate being more available in the lower part of the soil, it grows. If you see the root system's architecture, it grows a lot and it grows more towards the bottom side of the plate. And that means in this case that the plant is the shoot or the leaves, they're quite well developed. And this plant will also produce lots of seeds. And actually, you see here already we have the flowers. So it flowers quite early and it produces a lot. Whereas in this case, in the mutant, the root is not able to sense the nitrogen availability. So it doesn't really grow so much in search of this available nutrient. And that means also in this case, the shoot or the leaves are quite small and they are not yet producing flowers and soon the seeds. After we have this time course experiment, so we take scans of these roots after several days. So for example, 7, 10, 14, 17, 21 days. And then after you measure those, after you take those scans, you have to find a way to measure. You could measure with your own hands, right? With the aid of some computer-based software. However, it's quite demanding, it's quite time-consuming. And these days, with machine learning available, we scientists are also making use of machine learning a lot. And so what I can show you here is that these are the actual photos that I took. And then we use a software to try to detect automatically what is a root and what is not a root, what is the background. And once the computer can detect what's the root and what's the background, it can produce a different type of image, and then you can throw into a different software that can measure. So, for example, you can measure total root length, but you can also try to find out the area, root network area. So again, this plant where the gene is not functional, in this case, the root also grows more, just like the wild type plant. So the root grows more down and it's more efficient in taking the nutrient. While the loss of function plant, the root grows more to the sides because it cannot sense so well. It cannot move down as a gravitropic response. And because it's defective in this, it cannot be so efficient in taking the nutrients.
Hi, welcome back. So there was something that Marina showed me, which was the way you prepared the seeds that turn into the plants that you showed later. And I think it's an interesting process. Would you like to explain how you did it?
So usually, like in a plant lab, we often use agar plates. That means Petri dishes with agar, where the nutrient solution, so the different nutrients that we add, they are added to this jelly, to this agar plate. And I started my experiment there. So that's where my seeds were growing from the beginning. And the seeds were actually growing in agar plate with ammonium only as a nitrogen source. So they didn't have the best growth condition from the beginning. They were stressed. So in order to induce the stress, to push for the stress, that's what I did. I grew them only containing some small amounts of ammonium as nitrogen, and that's not ideal for Arabidopsis. And then after that, so we put them in the vertically. That's the benefit of these square plates. is that you can actually grow them vertically, and then of course the roots, they always grow down, I mean towards gravity, so that's why we use these vertical plates. So from there I started my experiment, and then you're gonna see you saw in the video the transfer, right, to soil.
Yes, exactly. So I'm curious, the reason why you induce this stress
Yeah.
in the seeds is to make sure that the roots grow just vertically?
No, actually
Oh.
the reason for that is because of this gene that I'm looking at.
Oh.
Yeah, it is a stress responsive gene, and that's what we're trying to understand.
Ah,
So it's
very
something a
cool.
bit more complex, but that's the point. Like in nature, sometimes some plants, they are more adapted to stress. Some plants, don't like stress at all, and they don't survive under stress. So in order to find out which genes are involved in this, I put them on purpose in a stressful condition to start the experiment. And what I was trying to see then in soil is how they recover from this stressful condition.
Very good.
Yeah.
So one thing that I think is amazing about your research is that you can understand how plants perceive the nutrients from the soil. So
Yeah.
one of the implications from your work can be to later on create guidelines for farmers to use fertilizers in a more responsible way. Can we say that?
Yeah. So it's already known. So I'm not the one who is like inventing this for the first time. It's already known that usually when you add fertilizer, of course the farmer adds because they want the plant to grow more, to produce more. But it is known that most of the fertilizer added in farms, especially if it's inorganic, it will remain in soil. So the plant cannot take it up. So the plant has some limit, and it will not take more than its limit. And so what we're trying to do for once is once the farmer is able to add less and have the plant that still grows more,
Exactly.
that's the goal, right? To help. So usually how this is done is through plant breeding, right? Because once we know the genes involved in this process, we can try to find plants which either don't have this gene functioning so well, or they have this gene that functions much better, it's more efficient. And then you can select for that through plant breeding normally.
Very well. So last week, on the 18th of May, we had the International Plant Fascination Day. In your opinion, why is it important to study plants?
Okay, so important, I don't think it's just important, right? I think plants are really the reason why we are alive on this planet, right? Because they actually give us the oxygen that we need to breathe. They remove the carbon from the atmosphere. They give us food, they give us shelter, they give other animals shelter as well, right? And so I think, it's not only important, but it's fundamental. And especially if we think now about this climate crisis, they also create cooler environments. So if you see that more and more in urban environments in cities, we have heat islands, right? So it becomes too hot because there's just too much concrete, too much construction. And when you simply add some, at least you make sure that there is a park near those areas with tall trees to give shade, to make sure the environment keeps cool, then it's also more pleasant for life. So it's not only, it's a basic requirement, I think, for life on Earth.
Absolutely. Now, in order to be the researcher that you are today and to be able to do the cool experiments like the one that you showed us, first you needed to become an expert in biology and more specifically in plant biology. So when did you realize that you wanted to be a biologist?
Okay, so as I mentioned in the beginning, I feel like I always wanted to be a scientist, right? Since I remember, I don't know, like I was 5 years old, 6 years old, I was already playing with this like little chemist kits. I was trying to find out my favorite book were like about the whys of nature, of life and nature, why things happen this way. Also, I really remember having this really nice book about astronomy. And I also had another book about human evolution. So none of them were like particularly about plants. But I was always into science. And I only got interested in plant science when I went to university. So I did biological science at university. And then that's when I became very interested. For once, it's in the countryside, so I'm a city girl, and then I got more connection with like being in the countryside of my hometown in Brazil. And also, there was a lot of lectures on very specific topics on plants. So we had plant physiology, we had botany, we had plant anatomy, we had economic botany. We had so many things and a lot of practical classes, and that's when I became very interested. And also one detail is that I realized by then that I would not be able to do human research, animal research. I just couldn't handle seeing blood, seeing like tissue, animal tissue and stuff like that. I was like, no. So I felt like plant, they really made me happy back then, and I started working with plants in 2004. And so it's been, 21 years. It's crazy.
Very well.
Yeah.
So you just said that you did your bachelor's in biological sciences, correct?
Yeah.
And walk us through your path after that.
So I then went to masters. Like it's very typical path in terms of course, if you're in the academic research, you have to just follow this path of doing master's PhD, right? So I did a master's still in Brazil. I started looking into how plants fight or mount their defense against pests. So I thought that was quite cool. And for my PhD, I decided I wanted to get out of Brazil. So I decided I want to have better working conditions, better salary, and also better living conditions. And that's why I decided it was the time, right time, when you look for a PhD. So I ended up going to Australia, which was a dream to me. It was really amazing. Really amazing place to live. And that's where I started my PhD in Perth, Western Australia.
That's really cool. So it also allowed you to know a completely different part of the world.
Yeah, and different people, like especially because Australia is very international, and it's very much also becoming very connected to Asia. So there's people from many different Asian countries, which I had never had contact before. And so I felt like that was a very enriching experience to me to move. So I recommend, if whoever wants to do science, I really recommend that you also move out of your comfort zone. It's really nice.
Absolutely. And what do you think that was the biggest challenge that you faced to be in this point?
So that was exactly the challenge as well, because I moved to a completely different side of the world, and everything was already difficult, of course. But also my research became quite complicated. I was working with this pest, which is Phytophthora, and it's very devastating for plants. Even in Europe it exists, but in Australia it's a massive issue. And it turns out that once you work with experiments, with living things, it's already difficult, right? So getting experiments to work is sometimes a nightmare. But getting that bug to work, getting the bug to do what I wanted it to do, it was pretty much impossible. And so it became technically very complicated. I started having issues also with what I wanted to do, what my supervisor wanted me to do. And so I decided it was not right and I would quit, so I quit. And how I managed to overcome it, right? So I think that's the most important, is that I did not take it personally. I didn't feel like, okay, I'm a disaster, like I failed. I thought this situation is not working, so I would just move on to do something else. And luckily, at that time, I also got offered to try to apply again in a different university. So I moved to Melbourne to restart my PhD. And I did, and it worked out. So, and that's when I started to work with also more like directly just plant and nutrients.
Very well.
Yeah.
There is always a project that fits us better.
Yeah. I mean,
yeah.
Yeah, so that's it. I think that's the message also for people who want to do experimental research.
Exactly.
Is that sometimes it can be very complicated and sometimes it's not your fault. Sometimes you just need to find another alternative and quit that project for now and then just find something else that for some reason is working better and gives you more results.
Absolutely. So, on the contrary of this, what is your favorite thing about your work? What motivates you the most?
I think it's because it's so dynamic. Like I get to always be curious because there's so many intriguing questions every day, and this dynamicity as well. So I'm never just sitting down in front of the computer. Like of course you can sit down in front of the computer sometimes, but when you go to the lab, to me it's like a kid going to a playground. So there's so many different machines, so many different equipment, and so many different things I can do. So I think to me that's what keeps me going for so long.
Yeah, there are no two equal days, right?
No.
And what is next for you? What do you want for you afterwards?
Yeah, so I'm actually reaching the end of my postdoc time. And after so long as like doing scientific research, I'm actually thinking it would be nice to try something new. I'm not quitting on plants. I'm really a very strong advocate for plant research and for the importance of plants for us and for the planet. And I would like to continue with that, but more in a maybe industry setting. So I would like to try to apply my knowledge to something that either can help more directly with agriculture at the moment, or for example, some nature or plant-based solutions for some urban environment. So I think that would be what I would like to try. I don't know exactly which type of company, but those things are, yeah, I'm finding it interesting.
Very well. And now, if you could meet your teenage self again, younger Marina, this girl who was so much into science and liked all these different things from chemistry to astronomy to human health, which advice would you give her?
I think it's just don't be afraid to follow your dreams. You just, if you want to do something, just go and try. It will happen eventually.
I totally agree with that.
Yeah.
We should always go for what we want.
Yeah, of course.
And now, for our This or That segment, this time I want to know your preference about several subjects.
Okay.
So, are you ready?
Yeah, I hope so.
So I will just start shooting. Lab coat or field boots?
I think I was into lab coat more before, and now I like my hiking boots.
Very well. Data analysis or the discovery moment?
Discovery moment.
The aha. Organized chaos or color-coded precision? What kind of worker are you?
Color-coded, 100%, because I have synesthesia, so I need my colors.
Okay. Writing papers or giving talks?
Giving talks.
So more in the moment.
I prefer to speak than to write, that's for sure.
Working solo or in a team?
I think both. Both are important. I like both at different times. But I can 100% do my job. And in science, that's the point. I think that it's nice to be flexible.
I agree. And would you prefer explaining your science to a classroom or a celebrity in a red carpet?
To the classroom. I'm quite happy. Yeah, I've done this a lot before, and I'm quite happy to talk to, especially like teenagers or like younger kids.
Very well. And now, for last, a bonus question.
Yeah. Okay.
Would you rather have your theory proven, but by a rival?
What do you mean?
By a scientific rival?
Or
to never proving your theory, but at least you learned so many ways it didn't work?
I think I would rather have it proven by someone else, of course. Yeah, I think that's fine. I don't have rivals at the moment. I don't think so.
Okay,
But
very
anyway, I think it's important, the progress of science, regardless of the person who does it. Yeah.
Very well.
Yeah.
Well, thank you so much, Marina, for
Thank
being
you.
here today, for sharing with us how you became a plant scientist and a bit about your very cool work. And I have just a little something for you.
Thank you so much.
Our Vista notebook, so that you can maybe take notes about your experiments. And also, for being such an awesome guest, I would like to also offer you the opportunity of getting a tour to the soon to open Vista Science Experience Center. It will open in early October, and you can very soon know more about it as well.
That's awesome. Looking forward. Thank you so much, Anna.
Thank you. And to our viewers, thank you once again for joining us. If you have any thoughts or questions, please leave them in the comments. We would love to hear them. And the rest, you already know. Follow us, subscribe to our channel, and stay tuned for more on the next generation of science, one awesome guest at a time. Cheers!
Bye.